So the first thing that we're going to talk about is the hydropathy plot. So this is what it, oh my gosh. So this is the hydropathy plot. And it's also called, the Doolittle algorithm, but they basically, what this plot does is it predicts the function of proteins. So it looks at about 20 amino acids at a time, and it sees how much energy is soluble in the water. So below 0 means it's hydrophilic, so it loves water, and above 0 means hydrophobic, it requires a lot of energy. So it looks at specific regions of the amino acid structure for stretches of 10 to 20, but these vary. And they see that if it's hydrophobic, these may be regions that are membrane domains of an integral membrane proteins. So what do we mean by that? So we mean that if it's hydrophobic, if it is above 0, it means that these regions are membrane domains of integral membrane proteins. They might have passes. So they predict function of proteins based on their structure. So what the hydropathy does is it sees how much energy is required to solubilize a segment of water a segment of protein in water. So the positive number means that there's a lot of energy that is needed because that segment of protein is protected from this hydrophilic properties of water, whereas the negative number means it's highly soluble. So always remember that when it's a negative number, when it's under 0, it's soluble. When it's above 0, it's not very soluble, and it requires a lot of energy because the protein is protected by hydrophilic properties. So remember that for the future. What else? Then we have freeze fracture. And what freeze fracture is, and we're going to write it down right here, freeze fracture. And what basically freeze fracture is, it freezes cells in liquid nitrogen, and then you smack it, and the lipids part separates, and you can see things embedded in the protein. So there's a plane of fracture between the bilayer, and you can kind of see the transmembrane proteins that are embedded in it. It's actually very, very interesting. But the only limitation for this is size. There's specific size that you can and cannot do. But just remember that for freeze fracture, you freeze it in liquid nitrogen and then you smack it, and then you can see all the transmembrane proteins. And then the next thing that we have is red blood cell ghosts. So red blood cell ghosts. Now what's red blood cell ghosts? Red blood cells ghost is when you take a cell, red blood cells, for starters, they don't have any nucleus. They're enucleated. So red blood cells, for a fact, they don't have a nucleus. So they are perfect to examine how cells work. So what basically happens is you can break it and everything leaks out, and you can study the membrane. So it's really, really important. So what happens is there's a hypotonic lysis that happens where we break apart the cell, and we wash it, and then we disrupt and we seal it, and then we can start seeing what the membranes are and all of that. So basically what the leaky ghost is when all the stuff leaks out of the red blood cell. And what we can do is we can usually put alpha helixes inside of the red blood cell, and then we can start seeing what's going to happen. So there's a lot of different things. There's radioactive, or we can radioactively or fluorescently we can label the red blood cell ghosts, and we can degrade them with enzymes and all of that different stuff. Now the fourth thing is liposomes. Now what the heck are liposomes? So this is liposomes. So liposomes is a little bit easier because you can make synthetic lipid bilayers, and we can insert proteins inside and see what's going on. But again, the limitation is size, because the size of the protein is very important. So with liposomes, you buy phospholipids and you make your own. You put them in aqueous solution, and they're aggregated, and we start using them for experiments. And we already know that lipid bilayers form spontaneously in aqueous solutions. So that's it for liposomes. So that's pretty much what happens. Now, the most important one is detergent solubilization. And what detergent solubilization is when you have some. So detergents are basically, now what is a detergent? A detergent is an amphipathic molecule that disrupts membranes. So they're ionically charged. So the SDS is one of those phospholipids that are very harsh, and they will disrupt membranes. And then we have non-ionically charged, such as Triton. So remember, for ionically charged, we have SDS, which is very, very harsh, and then we have non-ionically charged, which is Triton, and it's a more gentle kind of detergent, and it can be used to solubilize particular compartments of the cell. So why do we use detergents? We use them so that we can separate proteins from lipids for analysis. So their main function is so that we can separate them, so we can analyze what's going on in the cell, and we can analyze the isolated proteins in a more controlled environment. So we kind of just isolate the proteins using these detergents, and then we can study them. So that's pretty much what happens with detergents. So they purify proteins away from lipids, and they're hydrocarbons. So once that happens, when these detergents are used, they start forming micelles. And what happens is, once you isolate a protein and you place it into an artificial membrane, that's what we talked about with the liposomes. When you isolate that protein using detergent solubilization and you place it in the liposome, you look for protein localization, you look for functions, interactions with other proteins, etc. So using detergent solubilization is very, very important. So you can use, there's many different ways for you to understand how this works. So you can use centrifugation or column chromatography there's two types. So there's centrifugation and column chromatography. Both you can use to separate lipids from the proteins, and you can place isolated proteins in liposomes and look for localization and function and all of that. So different speeds of the centrifugation determine how big or small that phospholipid is. So SDS coats proteins with detergent, so that's negatively charged, and the proteins migrate through a gel to negatively electrode. So separation based on size and sometimes on weight and on shape, that's very, very important. So basically the SDS page that's what it's called, the SDS page electrophoresis. And what that basically is when the SDS coated proteins with the detergent, they go inside this vertical gel, and the proteins will be migrating through this gel. So that's what happens with that. So that's pretty much it for the membrane lecture. All right, bye.